Good afternoon. Thank you for the opportunity to present my work at this workshop in the beautiful city of Hangzhou today. I am Maria Depres from King's College London, and today I will tell you about my work on diffusion MRI of the fetal brain. Here is the outline of my talk. First, I will discuss artifacts in diffusion MRI of the fetal brain, focusing on motion and distortion. Secondly, I will talk about reconstructing white matter fiber directions from scattered diffusion signal. And finally, we will have a look at microstructure in transient layers of the fetal brain. We acquire diffusion MRI of the fetal brain to study developing microstructure and connectivity. In the top row, you can see fiber orientations of the white matter tracts, while in the bottom row, you can see the tracts themselves obtained using whole brain tractography. Diffusion MRI of the fetal brain suffers multiple challenges. First of all, fetus is moving and mom is breathing, so the data is corrupted by motion. Data is also corrupted by geometric distortion thanks to inhomogeneity of B0 field. There are multiple intensity artifacts including B1 inhomogeneity, spin history, and within slice motion. And finally, the diffusion signal is scattered and needs to be reconstructed on a grid. Once we addressed all these issues, we are able to estimate the fiber directions from the reconstructed diffusion signal similarly as we would do for adult studies. In this talk, I will only focus on motion, distortion, and reconstruction of the scattered data. So let's see how we can correct motion and distortion. So this motion correction strategy for fetal MRI has been around for a long time. The main idea that we acquire one slice at a time, because this can be done very quickly and this effectively freezes motion in time. So we will end up with several stacks of slices. However, we will see motion between the slices, as you can see in this picture here. Then the next step will be volumetric registration. We will select a template stack and all the other stacks will be registered to that. And after that, we can reconstruct the scattered data and obtain the first volume estimate of the volume. Of course, this requires a selection of a reconstruction method, but I will come to that later. And now that we have a volumetric target, we can register one slice at the time to refine the motion correction. And this can be done several times to refine the motion, and the registration is always rigid because we're dealing with the fetal brain. Diffusion MRI is acquired using echoplanar imaging sequence. This sequence is very fast but also suffers from geometric distortion. Our data is therefore corrupted by both motion and distortion. Solution to that is to jointly estimate motion and distortion using image registration. So motion will be estimated using rigid slice to volume registration, as we shown on the previous slide, while distortion will be estimated using deformable volume to volume registration with physically inspired constraints. And then we can iterate between motion and distortion correction step. Distortion is proportional to the main B0 field, and inhomogeneity of the B0 field is generated by interfaces between air and tissue. Here you can see a gas bubble in a maternal gut, and this bubble causes this distortion here in the image of the fetal brain. Another important thing to realize about the distortion that it happens only in phase encoding direction. So that's this direction in this image. The phase encoding, it doesn't happen in the two other directions. And this is the Y direction of the slice. And the fetal head is surrounded by water, not by air, so there are no internal sources of the distortion. It's from outside, from the maternal body. That's why the B0 field is smooth and obeys Laplace equation. So that means that it should be easier to correct actually than in adults or neonates. So here is how we design our deformable registration for distortion correction. As a target, we will use a structural theta-weighted reconstructed volume that doesn't contain any distortion, and we also assume that we have motion parameters that were estimated using slice-to-volume registration. So we will use this structural volume to simulate the undistorted slices. So first we position the volume in the scanner space according to the motion parameters that we estimated, and then we apply point spread function to simulate the undistorted slice. And then we can correct the distortion in the acquired EPI slice by deformable registration in phase encoding direction only. And also we apply Laplacian constraint on the deformation field. And here you can see some example of what happens if we use a Laplacian constraint or not. So this is a acquired field map, which is ground truth for us, and you see that with Laplacian constraint we have nice smooth field and it corresponds to the acquired, while we see some artifacts if we don't apply the constraint. And these artifacts would be even more prominent if we didn't restrict the registration to phase encoding direction. The distortion was estimated using the non-sensitized B0 stacks, and in this example you can see how the distortion was nicely corrected with this method. The same distortion field can also be applied to diffusion sensitized stacks, as you can see in this example. So I have presented a registration-based method for distortion correction of fetal brain EPI. Its main advantage is that the distortion can be estimated directly from the data. The disadvantage is that it might not capture well variation of distortion over time, but overall it's a good solution for off-the-scanner acquisition of fetal diffusion data. There are alternatives. The traditional one is acquired field map, but this needs an extra scan and might be difficult to use correctly. Or what we have done in DHCP is to estimate the distortion from the phase of gradient echo scans. The problem here is that diffusion data is spin echo EPI, so we needed dual echo tailor-made sequence implemented on the scanner. So the first echo is diffusion data and second echo is for estimation of distortion. If you are interested in my registration-based distortion correction software, you can get it from my GitHub repository, and you can also email me if you need any help. Let's now move to the second part of my talk. How do we reconstruct the DMRI data? So far, we have estimated motion using rigid registration of slices to reconstructed volume, distortion using deformable registration of B0 stacks to stacks simulated from T2 weighted data. We can also correct B1 bias field using standard techniques such as M4. What we need to do now is to reconstruct scattered diffusion signal on the regular grid, and once we've done that, we can estimate the fiber directions using a standard tool such as MRTrix. Diffusion signal is a function of spatial location and sensitization gradient direction, and for its spatial location it is therefore a function on the sphere. You can see an example of diffusion signal in the fetal brain here. Because it's a function on the sphere, it can be parameterized by spherical harmonics representation. Here you can see the spherical harmonics basis function, and if we take a suitable linear combination of this basis function, we can approximate any function on the sphere. Another important thing to realize is that diffusion signal is orthogonal to the white matter fiber orientation. So it's because where we have diffusion, we lose signal. So to obtain the fiber direction, we can use constrained spherical deconvolution. Diffusion signal in fetal brain is scattered, which means not on the grid, and this is due to random fetal motion, and it's scattered not only in spatial location but also in gradient direction, because the gradients will be rotated by the rotation component of the fetal motion. So how do we obtain the spherical harmonics representation of the diffusion signal on a grid? To do that, we first model the process of acquisition of the fetal diffusion data. We start with spherical harmonic representation on the grid, which we do not have but want to estimate. We then sample the spherical harmonic functions at the rotated diffusion gradient and obtain the diffusion-weighted 3D volume of the fetal brain. Now we can apply motion parameters to position the fetal head in the scanner space and then apply point spread function to simulate a slice. To estimate the spherical harmonic representation of our diffusion signal, we aim to minimize the difference between the acquired and simulated slices. This is the only equation I will give you in this talk to explain this process better. So what we try to find is the spherical harmonic coefficients C. And when we calculate the linear combination with the spherical harmonic basis functions, which are y, and sample it in the gradient direction g, we will get the 3D signal for the gradient G, which is the diffusion weighted volume. Now if we apply the spatial point spread function, we will get the simulated slice, and then we can compare it to the acquired slice as by minimizing the sum of square differences. And we can find the spherical harmonic representation or these spherical harmonic coefficients using iterative optimization methods such as gradient descent or conjugate gradient descent. Here you can see the resulting white matter fiber orientations for the fetal brain of various gestational ages. We can also observe crossing fibers. This technique works for both standard and DHCP acquisitions. The limitation is that it works only for a single shell, which means a single p-value. It could be 1000 or 750, for example, and DHCP uses a multi-shell extension of this technique called shard. If you are interested, the software is available from my GitHub repository or also from SVRTK repository. And of course, email me if you have any questions. Finally, I will briefly describe our recent study of microstructure of transient layers in developing fetal brain. This study uses fetal diffusion data from the Developing Human Connectome project and was done by Sean, my PhD student Helena, and my undergraduate student Ami. First, we use the reconstructed diffusion MRI signal to feed two different models. It's the diffusion tensor model to extract fractional anisotropy and mean diffusivity, and then also multi-shell, multi-tissue constrained spherical deconvolution to extract tissue fraction and fluid fraction. Then we combine this map with a segmentation of transient layers. This is a CNN technique that was developed by Helena. And from that we extract the average regional microstructural measures. We found that fluid content measured by MD and fluid fraction is increasing up to 28 weeks of gestation and then decreases. And this was the case for all layers. However, the peak of fluid content was at different time points for different layers. The tissue fraction showed an opposite trend, which makes sense, decreasing up to 28 weeks of gestation and then increasing again. Interestingly, tissue fraction seems to be only increasing in cortex and is minimal in different layers at different time points. And finally, Let's have a closer look at fractional anisotropy and tissue fraction in cortex. They show opposite trends. While cell density, as measured by tissue fraction, is increasing, the FA is decreasing. And this can be explained by loss of radial orientation of the fibers in the cortex. You can see it here. We can see the fiber direction being nicely perpendicular to cortical surface in young fetuses, but as the development progresses, this feature disappears. And indeed, in adults, there is no directionality in cortical plate. Finally, I would like to thank Professor Joe Heinel and Dr. Donald Tournier, who significantly contributed to the methodology presented here, and also to all my co-authors and the Developing Human Connectome project. I would like to thank to my amazing group, Baby Media, who work on a variety of the projects on perinatal MRI that I did not have time to mention, all my colleagues in the Center of the Developing Brain, our NHS Trust, and in particular St. Thomas Hospitals that allows us to acquire such amazing images, and finally to our funders. And thank you also for your attention.